And we are looking at the purines that have a two-ringed base. And we've made one of the rings, but we're continuing to make the second ring. Now, in most of these cases, as we are adding carbons or nitrogens to the ring, we've been doing them one at a time, one atom at a time. The only one that added more than one was when we added glycine. And so we completed last time the reaction that phosphorylated the carbonyl. And so the acid itself. And we know that when we add an acid, this activates the ketone. And so if I attack here, this now becomes a good leaving group. And so what we are going to add to this ring system is a nitrogen. And we are going to get the nitrogen from aspartic acid, and we're going to take the nitrogen from the N-terminus. And so when I look at the beginning, carbon attached to a phosphate, now carbon attached to a nitrogen, nitrogen having one hydrogen, we have a nitrogen that has two. So I will take a base and take off one of those hydrogens. I now have a negative nitrogen. That becomes a nucleophile. Attack the ketone, up, down, and this is the investment of the energy, the phosphate coming off. Okay, probably should have an arrow that says Pi coming off, and I have this entire aspartate added to the growing molecule. Now, again, I only wanted the nitrogen. And so I can do one more reaction and have fumarate come off. Okay. If you take a look at fumarate, what pathway are we in? Citric acid cycle. And so we are coming back to all of the pathways we've worked before, and so they're all connected. If I take a look at this in inventory, acid, acid, carbon. How many hydrogens are on this carbon? One. There's one. Carbon. How many hydrogens? Two. And then the acid. And so I know where my extra base is. All right. And so I will have fumarate as the leaving group, and I will leave a nitrogen. Now, the reaction going from here to there with a fumarate leaving group, how would you classify that particular reaction? Which one is that of the five that you know? An E2, because it's eliminating, creating a double bond as part of that process. All right, all that work, it seems, just to get that one little nitrogen. All right, the next, we're going to add a carbon. And remember, THF is a carrying molecule. It's much larger than we show, but the two nitrogens that are involved in carrying methyl groups or carbons that are in other oxidation states are the 5 nitrogen and the 10 nitrogen. We're actually going to show at the end of this chapter one that is carried between the two. So in one case we saw a methyl sitting here and that was transferred. In another case we saw the formyl group being carried by the nitrogen, and we're doing that again. All right, if I look at the reactant and the product, the only difference happens here. Nitrogen 2 hydrogens, nitrogen 1 hydrogen, and the formyl group. Kick up, kick down, and picks up a proton. This is similar to a reaction that we saw previously. All right, we have now a ketone at the top, a nitrogen, a ketone at the bottom, a nitrogen, and if you take a look, we have 1, 2, 3, 4, 5, 6 atoms on this side that can make that 6-membered ring. Our 6-membered ring will actually react, and if I take a look at this, it's an internal attack. I see this ketone, this nitrogen has 2 hydrogens, only has 1, so I have to take off one of those. But this nitrogen is now attached to a carbon that is a hydroxide. All right. And I don't have a hydroxide over here, but I have something that can become a hydroxide. When we attack a carbonyl, which we do a lot, there are 3 different things that can happen. One is kick up, kick down. Okay? You would find a ketone in the product. The other is I attack and I can pick up a proton. That second movement results in a hydroxide. The third thing that we have seen is when I attack, the electrons kick up. Instead of picking up a proton, it can attack ATP to become phosphorylated and activated. That happens a little bit more rarely, but that is one of the 3 main things that can happen. And so I'm now at this particular structure that has a hydroxide, and I've made the 6-membered ring bond. Now, if you take a look at this, we are going to gain stability if we could cause resonance. And so to get a base pair called IMP, we can take a look and see if this hydrogen, which is absent here, and this hydroxide, which is absent here, are allowed to react. I create a double bond in the ring system, which is going to lead to stability, and we're leading to the reaction or the products. Now, the 2 purines that we make, okay, IMP is going to be able to go 2 different directions. What are the 2 nucleotides that are the purines if we have the triphosphates? So it's something TP and something TP. What are the 2 base pairs? ATP and GTP. IMP is a intermediate between both of those. Okay. IMP will also be, we're going to see, the degradative intermediate when both of those degrade. All right. Up here, I'm being lazy. GTP, I wrote out GDP and just the phosphate at the end. Okay. So 2 phosphates plus 1 more makes 3. All right. And this is really kind of weird, this reaction. All right. What actually happens in this? This one's a little bit different than we've seen. Okay. No changes around here. Changes around here. Okay. How in the world do I get this double bond from this structure here? Does anybody have an idea? Correct. In the formal Lewis structure, there is a lone pair there. If you look at this, in the Lewis structure, there is no lone pair. So that movement of that lone pair creates the double bond, meaning too many bonds to carbon, so the electrons will kick up onto the oxygen, and then that becomes a nucleophile and attacks the GTP. And I kick up, and I kick down, and I kick out. Star. I am in the process of making AMP. Okay? To power that, I use GTP. Okay? You're going to find if I'm going to make GTP, what's going to power that is AMP. So they are balanced between the two. All right. I have now phosphorylated that intermediate. Usually we do that so that we have a good leaving group. And I'm going to use aspartate again, but in the aspartate case, I really want to just extract that nitrogen. But to do that, okay, I'll take this hydrogen, I will attack here, and this is weird, okay? I will kick up the lone pair. All right, that doesn't get me to my final product. What has to occur, and I'm going to use a different color, in a second reaction, a base will take this proton, reform the double bond, and the leaving group off of that deprotonation is the pi. Okay, almost there, I mean right on the edge. We will have fumarate fall off again. And by taking that particular proton and creating a double bond, and this picking up a proton, which we've seen just at the beginning of this lecture, we have fumarate in the citric acid cycle and AMP. Okay, spent all that time putting each individual atom in the ring, formed the ring, and we now have AMP. Coming back to IMP, all right, I told you that this was the one that could go either way. We're now going to create GMP. The enzyme becomes part of the reaction. This is also a oxidative reductive, a redox reaction. We have NAD+. The enzyme is going to have a reactive cysteine. And if I take a look at this, the cysteine has to react to this. It's there. Then I get a whole series of changes of where the double bonds are. So I attack the carbon to make the bond that I need. I have to move the electron pair to match the product, which then makes this one move, which makes this pick up a proton. By picking up the proton, I create a much more stable intermediate than if it was just the negative charge. All right. Now, the next reaction requires NAD+, so I'm going to have a hydride leave. All right. When I look at this, all right, that doesn't move. The only hydrogens I have is this one and that one. One of these is going to be the hydride that leaves. And just so happened I put the NAD right close to, where it needed to be. So the base is going to take that. I will reform the double bond. I then have to take the ring electron pairs and put them back. But now the leaving group is the hydride. I'm still connected to the enzyme bound through a sulfur. Okay. Now, to get to the next intermediate, I have a hydroxide. Base will take this, attack where I need the hydroxide, and again, have an intermediate where I have the electrons move around the ring. All right. This is where I am done with the enzyme. So the sulfur needs to be the leaving group. And that becomes the cofactor, or the amino acid that is covalently linked. The enzyme is ready to attack again when this product leaves. Now the last reaction, all right, there is an equilibrium between these two, and so what we're doing is we just have an isomerization because hydrogen is there, hydrogen moves, oxygen is there, oxygen, we don't lose or gain anything, we just rearrange. And so okay, and we get xanthine, or xanthine. Not the base pair that we thought, but we're almost there. The last is for the nitrogen to be added to get to GMP. To do that, it is powered by ATP. And so here, the investment to make GMP is powered by ATP, which is the reciprocal of what we just saw. In this case, if I deprotonate this, form the double bond, this can attack, kick up, kick down, and ADP is the leaving group. I have an activated intermediate. Last time we used aspartate. This time we're going to use glutamine. To provide the nitrogen. And what has to happen here is this kicks there. I'm now attacking a carbon that has a double bond, so the intermediate will have the nitrogen negatively charged. It will reform the double bond, and the phosphate is the leaving group along with glutamate. And I have GMP. So I have ATP and GMP, or sorry, AMP and GMP right now. Now, the pyrimidine synthesis in terms of knowing where everything comes from is a little bit more simple. The aspartate, okay, is a derivative from the citric acid cycle, and the carbamylyl phosphate is in a pathway that you have seen before. Do you remember where that one is? Urea cycle. Also, aspartate can actually come from the urea cycle also. So, we have aspartate and the carbamylyl phosphate. And so what in the world happens here? Okay, in the product there's the nitrogen with a ketone and a nitrogen. Well, here's the nitrogen, there's a ketone, and on this side I need a nitrogen. That will be this one. Up, down, and out. Okay, phosphate leaves. I'm almost at the ring structure right now with that one movement. The next reaction has cofactors inside the enzyme. They are zinc, because we have lots of negative charge sitting here. That acid has a negative charge that is resonating, and then when I do my next reaction, I'm going to have an additional negative charge. Those lone pairs of electrons can actually coordinate to the zinc to stabilize them. And so this allows me to do a reaction that looks like it is very high in energy, having lots and lots of charge. Okay, there's the stable intermediate. And in this case, we will then have it form the double bond. This will pick up a proton for hydroxide to leave, and then as soon as the hydroxide gets into the buffering system, it will create water. All right, I have a 6-membered ring. Now, here's something different in the pyrimidine synthesis. We started with, in the purine synthesis, the ribose. And we added a nitrogen, and then we built the ring base around it. Here, we're starting to build the base pair without the sugar yet. Okay, so it's kind of backwards between the two. FAD is an electron carrier, and so I can create the elimination that is found in the product by taking off these two hydrogens. All right. Now, based upon what you know, the hydrogens could come off of this particular ring system, that particular carbon, or the hydrogen can come off of this particular hydrogen. Which is more likely the one to be deprotonated, blue or green? I know it's 50-50. Green, blue. Okay, so green or blue really doesn't matter to me. Why green or blue matters to me? Yeah, because. That's what I like. Because. One is alpha to a carbonyl. One is alpha to another carbonyl, but they're different. One is alpha to just a carbonyl, one is alpha to an acid. Okay? If you are alpha to a carbonyl, then if you take a base and you take that off, then you have this resonance. Okay? That stabilizes it. If you are alpha to an acid, the only thing that you get is a negative charge there. Why can't you get a resonance structure? It really looks like you could just say, oh, I can push this down here, right? And that can go up there. But when you write an acid this way, does it look like this? No. This is already resonating back and forth. Okay? And so it's already in resonance, so I can't enter that in. The other reason is, if by chance I put the negative charge here and I put a negative charge on this, I've got two negative charges very, very close to each other. That's even worse in terms of energetics. So being alpha to a acid doesn't make it acidic. Being alpha to a ketone does. Okay? And so most likely, the base is going to take this, the electrons will fall in, and we'll take a long route to get to the FAD. And then the FAD has to pick up a proton. All right. Still haven't got to where I want to go, but I'm closer. All right. The slide is different than in your notes. I just looked up a reference, and there is a new mechanism that is supported, although there's debate about this particular mechanism. Yes? Say that, the other one? Oh, sorry. There it is. Okay, good. When I looked up the reference, there is competing labs that are trying to prove this particular mechanism. There are 3 separate mechanisms that are proposed in the literature. This one is the most supported by the data at this moment in time, but it's very, very strange. And so I will not make you responsible for this one, so you can kind of just watch this one because it creates something very, very strange. Here, the electron pair on the oxygen will form a double bond, and the pyrophosphate will leave. All right, this doesn't look like a wonderful intermediate. Positive charge on the oxygen and the elimination that occurred. Okay. The only reason why that this is stable is that there are a number of amino acids that are stabilizing this particular form of the structure. The second movement would be to take off this proton, to attack it, and then to allow these electron pairs to go back onto the oxygen. And so just kind of an oddball reaction, but it is a very heavily stabilized intermediate by the amino acids. A lot of times you have 2 or 3 amino acids in the active site that are actually interacting. When I was looking at this, there were 7 separate amino acids that their side chains were stabilizing this transition. And this is the best proposed mechanism for this. So it is complex. This one I wouldn't expect you to predict. All right. This mechanism kind of looks crazy. To create a CO2, I take the extra pair of electrons to form the double bond, and this pair of electrons will pick up a proton. All right. And I get UMP. Okay. UMP. Great. U. What kind of base pair is U associated with? RNA, right? And look at this. All of the things that we've been synthesizing have been RNA up to this point in time. Okay, once you have UMP, TMP, and we haven't got to CMP yet, but we will in just a moment, there are specific kinases that will take them to the diphosphate state. Then the enzyme adenylate kinase can convert all those dyes to triphosphates. Okay, and at the diphosphate stage, another enzyme called nucleoside diphosphate kinase can interconvert all the dyes to the tri. So we have 2 different enzymes that can move the diphosphates up to the triphosphate. Now, what is important here is that this is synthesized as the monophosphate. This is synthesized as the monophosphate. The adenylate and the GMP are synthesized as monophosphates, but we're going to see that some of these others have to be in a higher phosphorylated state. In particular, to get to the CTP, we have to use the UMP that we just synthesized and phosphorylated up to the triphosphate stage before it gets into the enzyme to make CTP. And so we are going to use ATP as the power. We're going to use glutamine to provide the nitrogen. And we are going to form a substitution where once was the ketone is now the nitrogen. We have seen this reaction before exactly with ATP going to ADP. Okay, and so the intermediate will have a phosphorylated oxygen, and then the nitrogen will attack, and we can form the product. Okay, so at this point in time, we have all of the RNA bases, A, G, U, and C. That is great. To react and create DNA, okay, all we have to do is take the hydroxide off and put a hydrogen there. It looks so simple, especially because you have NADPH. Okay? It is not that simple because it looks like you could just do this. Attack, fall off, pick up a proton, and that is not how it goes. Okay? Because all I need to do is have that hydroxide fall off. But it's much more complicated. The enzyme is very much controlled. Okay? All right. It is a dimer. It has a catalytic subunit that has some features of it. Reducing power. Okay. Also, a radical. Okay. So we've seen a radical reaction before, and when radical reactions happen, we get really strange things. This can work on the diphosphates. Okay. And so either it synthesizes a triphosphate, has to go to the diphosphate, or synthesizes a monophosphate, has to go to the diphosphate to be converted. Okay. And so the active site can bind all of them, but not at the same time. There are specificity sites. Depending upon what is bound here, okay, will cause the active site to bind one of these four. So there's a combination or connection between what's bound in the regulatory site and which one of those binds. And then primary regulation, meaning whether it goes fast or slow, designated by either the RNA ATP or the deoxy ATP, and the presence, trying to keep a balance. All right? Now, I'm going to show you this. No, I'm going to show you the mechanism. And the one thing I don't want you to do, I think I have a slide coming up. I don't necessarily want you to know which one of these causes which of this. What I do want you to know is that there is a control point at which I only do one at a time. Okay? But I'll show you the mechanism. The mechanism will not be something that you're responsible to reproduce, but I want you to understand what happens. All right, the radical is the first reaction, and we have to take the hydroxide off the 2 position. So obviously, the first reaction that occurs happens with the 3 position. Weird. And so half arrow for this hydrogen electron, and then 1 electron will go onto this 3' position. All right. We said we needed NADPH, right? But NADPH is going to be a hydride donator at the end. Second movement, this hydroxide is going to pick up a proton, okay, leaving a negative charge on the sulfur. This is a good leaving group. This is exactly how you would write it in an organic reaction under an acid-catalyzed mechanism. You make a good leaving group before it actually leaves. And what will happen is it will actually leave. These electrons will go onto this oxygen, leaving a carbocation. And so you've got a carbocation and a radical in this reaction. Strange. This next reaction, the sulfur comes in, creates a disulfide bond. Here's a hydride attack. Well, a hydride negatively charged to a positive charge, that seems logical, and I will cause that reaction to occur. I will then reform the double bond, or sorry, the radical onto the enzyme itself. And what floats out is this DNA. The NADPH will be used through a series of reactions to give the electron back to and break this disulfide bond into its reduced form. So NADPH will go through a series of reactions to give the electron pair back here. So we can go. All right, strange. What is also very strange is something else. Oh, the flow of NADPH. Usually it goes through NADPH to FADH2, FADH2 to thioredoxin, thioredoxin to the ribonucleotide reductase, so that we can make the DNA. Usually if one pathway in a mechanism stops, that totally stops synthesis of something essential. There is an alternative pathway going through glutathione. We looked at that before. In this chapter, it's at 3 amino acid intermediate through glutoredoxin that can also fund this. So fundamentally, we need the deoxyribose, and there are 2 pathways for that reduction to occur. Oh, this is the slide that you don't necessarily need to know, where we can talk about which ones are being produced. We start with U, C, G, and A, all the RNAs, and we make the DNA A, the DNA G, the DNA C, and this. What's wrong with that? Yeah, we don't have a U in the DNA. Okay? So that intermediate is not used anywhere other than in synthetic pathways. To make the T, we use the dioxy UMP, and so it was created as the diphosphate, has to be dephosphorylated for it to fit into the next enzyme. And what we're going to do is we're going to take the proton off, and add a methyl group. Okay? Well, to do that, we're going to have tetrahydrofolate carry a methyl. But it's in a strange way. This carbon of the methyl will come from a methylene that is bound between the 5 and the 10. All right? We will then get an intermediate that has to be regenerated. This will give the TMP that we want. We have to use NADPH to reduce this bond, and then we have to re-give the carbon coming from serine back ready to react. All right, this particular reaction is a very important reaction and one of the first that was co-opted to attempt chemotherapy. Okay, and so chemotherapy, to fight cancers, we have to try to find a pathway that will stop fast-growing cells. One mechanism that was thought about was this. If you don't have all of the DNA nucleotides, then you're going to stall the cellular division. And so we don't have to attack all of the synthesis of nucleotides, but if we can get one, that should fight or reduce reproduction in cancers. Okay, and so we want to stop the overproliferation of cells, and so we're going to stop this synthesis at both the enzyme that makes it and the enzyme that regenerates the substrate to make it. So here is the reaction mechanism. that's highly involved with the enzyme making covalent bonds. Here is the normal substrate, okay, the UMP. This is the chemotherapeutic agent, fluorouracil. Instead of having the hydrogen there, I have a fluorine there, okay? That's going to make a difference. Now, in the first step, the enzyme reacts by allowing a sulfur to attack. Both intermediates allow that attack to occur. Both have a negative charge on the carbon, that's fine. And both will do the second reaction, where the negative charge will attack the carbon. Now, there's also one other attack that occurs in the ring structure in the dihydrofolate that comes in. The negative, or the lone pair will kick down, and this will pick up a proton to actually have a leaving group of the nitrogen. All right? The next reaction, both of them will attack to make a bond to the carbon that I want. Okay? And that's great. The next reaction is a base is going to deprotonate. This electron pair will create a double bond, and the carbon will be transferred to the ring structure. This right here is a dead-end complex. Why? What's the difference? This base did what? Deprotonated, right? I have a base here, but what don't I have? A proton. Okay? So, I'm covalently linked into the middle of this enzyme. It is a dead-end product. We've also called this inhibitor of this enzyme a suicide inhibitor. Because it's become covalently linked, it can't come out, and this stops the synthesis of the thymine. To finish out the actual synthesis, I have to have a reduction occur. Electron pair that is working within the tetrahydrofolate will allow a hydride to leave to create the methyl group. Now, because it has a hydride leave, I then have to use NADPH to regenerate it, and that's the next step. Okay? The dihydrofolate back to the tetrahydrofolate is done by allowing NADPH to react with it. But what we do in chemotherapy is we block this particular enzyme also. We use these two drugs. The drugs right here, all right, would you consider them what kind of inhibitors? Just by looking at what you see in the structure, you may or may not know. Because I didn't show it to you a lot, but look at this. A 5 and a 10 nitrogen that are in arrangement, it kind of looks like the tetrahydrofolate, or the dihydrofolate. So it's got a similar structure. If it has a similar structure, then it's most likely a competitive inhibitor. It's going to bind into the same place because it's got basically the same structure. The enzyme really doesn't distinguish what it is by looking at it. It distinguishes, does it have the same shape? If it has a very similar shape, it will bind into the pocket. This right here doesn't necessarily look exactly like this one. This could be an inhibitor that is a competitive, but usually if you have a totally different shape, it may be a non-competitive inhibitor, and so it binds somewhere else, makes a conformational change to slow the enzyme down. So we may have both a competitor and a non-competitive inhibitor to slow it down. Okay? Now, the reason why we use multiple drugs is because there is a transporter in cells that will normally take out anything that's hydrophobic that's not supposed to be in the cell and pump it out. When we stress the body with these drugs, we get a lot of organic compounds inside these cells that aren't supposed to be there, and this sometimes upregulates that transporter. And we make so many copies of the transporter that no matter how much drug we give a patient, we never get what's called the therapeutic concentration in the cell, the concentration high enough to bind to the enzymes we want to, because this pump is working so fast. And so we tend to hit patients with very strong doses of a lot of different drugs, cocktails, to fight cancers, so that we can minimize this resistance.